Welcome to our third and final lab podcast on the digestive system. And in this session, we are going to look at the accessory digestive glands. The first that we will look at as far as the major glands that we have, we're not looking at the minor salivary glands, but we're going to be looking at the major glands. And this is a section through the parotid gland. It is going to be lobed in nature and practically purely serous. And so even at this level, you can see all of the dark staining tissue. You can see the lighter connective tissue regions, which have divided up into lobules, and even some slight pinkish color that are going to be the connective tissue septa, as well as ducts that you can see. It almost has the look of skeletal muscle to a limited extent, but as we get higher and higher magnification, we will be able to detect the acini that are present in the parotid gland. And here we can see those acini. We've got our ductal system. And here we actually have an intercalated duct. You see how it narrows? It's almost getting squamous in nature. At this point, this is also getting more and more narrow. Here we have our cuboidal shaped cells. This is going to be more of a cross section through that, and then we get out into our connective tissue. But notice these acini, purely serous, and that's going to be indicative of what we see with our parotid gland. Now as we move into another of our major salivary glands, this is going to be the submandibular gland. Again, looks very much marbled like we would see with muscle. We see more white or lighter color tissues here because for submandibular gland, our composition is going to be approximately 90% serous, but we will have 10% or so of mucus acini. So as we look at the field of parenchyma, the first thing we're struck with the number of ducts that we see in cross section. We can also see the dark blue to purple parenchyma, the serous, but then we see the lighter, almost aqua color. Those are going to be the mucus acini. And just this global view, looking at the percent of purple to this aqua color, you can see about the 90 to 10. Certainly overwhelming, we're going to have the serous. But then the number of these cross sections of ducts, these are cross sections through the striated ducts. And when you see that many, remember in submandibular gland, we have very long striated ducts. And so that's why we're going to see so many of these in their sections. Higher magnification again, we can pick out the purple color for our serous acini. Here we have mucus cells, mucus acini. Here we have intercalated ducts that are diving in to our acinus here in this mucus acinus. And then we have a lot of these cross sections. And even though at this magnification it's difficult to see, I think you can sort of see the radiating nature of the striations along the basal surface. These are the striated ducts. Now, this is one of the few sections I could find that exemplifies sublingual gland. Now, sublingual gland is going to be predominantly the majority of the parenchyma is going to be our mucus type parenchyma. We will have serous demolunes, and we can see some of those on the periphery here. We don't really see any serous demolunes. We have a lot of serous cells in this particular area, but predominantly we're looking at a mucus type gland, and that's how we'll distinguish parotid from submandibular from to this one, sublingual. Now we're leaving the oral cavity and we're moving into pancreas. So as we look at this, it doesn't look too dissimilar to what we saw with our salivary glands. We see the marbled look. This is certainly going to be much more tissue. It's a much larger organ. And we can see lighter staining regions here that may or may not be ducts, may or may not be connective tissue. So we want to see a little bit higher mag before we really decide what it is we're looking at. And as we go to this higher magnification view, we're going to see these light areas, these rounded areas that are present in this sea of serous parenchyma. This is all of our exocrine portion, parenchyma of the pancreas. And these are our pancreatic islets, or islets of Langerhans, which is going to be the endocrine portion of the pancreas and everything that's being secreted there. So with this view, if we had the accompanying inset and make it even easier, we're going to be seeing the islet cells and our pancreatic cells. Once again, very high magnification of the exocrine portion, looking at our parenchyma. We're going to have the acinar cells that are present here. But also remember, in the pancreas, we also have centroacinar cells, these lighter cells that we're going to find sort of lining and being present in the center of these acini. And so that's largely what we're seeing here. We also see a lot of blood cells. We see some polymorphonuclear leukocytes present in our capillaries. They're going to be nourishing this tissue, so we will see quite an extensive capillary network. Another view of the parenchyma present in the pancreas. Again, we can see the serous parenchyma. We have those centroacinar cells almost intercalating in centroacinar cells, so they're quite abundant present in this pancreatic tissue. Now we're going to move to the liver. And so in this low magnification view, the first thing that our eyes are drawn to are these large white patches that we see. Now the biggest ones, those are going to be some of our veins that are present within the liver. But as we look at a number of these small spots, especially the ones that you see with the inset, you're going to be able to see white radiating patterns radiating out away from these large white structures. These are our central veins, and we can see the hepatic sinuses radiating away. And so even at this low mag, we can have a hint that we are in fact looking at liver. So if we look at one of these in higher magnification, here we can see our central vein present in the center, our hepatic sinusoids radiating away. And in the vertices of our classic lobule, remember, we're going to have our hepatic triad. We definitely have a representative of the venous supply. One of these is going to be for bile and one for arterial blood. It's difficult to make out in this particular magnification. Here we have an adjacent triad. Here we have a triad in this particular picture. Now, as we look at the hepatic cords, these are going to be the hepatocytes that basically define the boundaries between sinusoids. So here we have a hepatic sinusoid. Here we have a cord of hepatocytes, a sinusoid, cord of hepatocytes, and we can recognize the endothelium lining the sinusoid. Remember, that's our sinusoidal type 3 capillary endothelial cells, and there we can have that as well. The lighter area beneath that is going to be our space of Dissay, and we'll see that a little bit better once we get to some EM pictures. Then we're going to have these cells that are present in the sinusoids themselves. Now, some are going to be blood cells. We're going to see red blood cells as well present in these sinusoids, but these large cells that have the vesicles present in them, remember, these are going to be our resident macrophages or our Kupffer cells. Again, we have a different view of our central vein showing the lining endothelium, our hepatocytes. Really, this is a typical H&E stain that we're going to be accustomed to seeing when we look at liver. Now, in the vertices of our classical lobule, we are going to see our hepatic triad. So whenever we look at these hepatic triads, the first thing we're going to see is the very large luminal diameter of the venous supply, so part of the hepatic vein. Then we're going to look for the arterial supply, and we can see our smooth muscle here, so this is going to be part of our arterial system, so hepatic artery. And then we're going to see our cuboid-shaped epithelium lining a lumen-bound structure, and this is going to be a portion of our bile duct. Now, it looks like we have some other vessels in here as well. These very small ones that we see, just endothelium, fairly large lumen. These are going to be lymphatic vessels. And you may or may not have more than one of each representative in this area of the hepatic triad. But the triad really is for the venous supply, arterial supply, and the biliary supply. Once again, we have our hepatocytes that we see with the cords of hepatocytes. We can see our lining epithelial cells, the endothelium. We have blood cells in the center, plasma cell, macrophage on the surface, likely another macrophage over here. But as we look in particular sections between hepatocytes, these are going to be our bile canaliculi that we see. Bile forms in the hepatocytes, transported into the bile canaliculi, and directed away from the central vein toward the periphery to our bile duct. Here we have another view, and in this case, the dark line that you see near the center, this is going to be a bile canaliculus. Now we're going to look at somewhat the architectural structure of the liver. We're going to be staining the reticular fibers with this bright black to orange type stain. So let's look at the reticular fibers and how they're laid out. That's going to be supportive of all the cells that we have in the liver. So we can recognize the central vein, and again we can somewhat recognize the radiating pattern away from that central vein that's going to define the position of the cords and the sinuses. And upon higher magnification, again, we see the reticular fibers defining the area of our central vein, and then positioning themselves so that the cells can stack on them as the hepatic cords, leaving these gaps between the areas where we're going to have our hepatic sinuses. Now we're going to move slightly peripheral from the liver. We're going to look at the gallbladder. It's going to be intimately associated, and remember, its adventitia is going to be part of Glissen's capsule of the liver that supports that intimate relationship between the liver and the gallbladder. So in this particular view, we can recognize hepatic tissue in the upper left. We have some vessels, we have our peritoneal covering, our visceral peritoneum, and then on this surface, we can see the epithelium that we have for our gallbladder. Submucosa, we can have some smooth muscle that's going to be present here. Remember, that responds to our cholecystokinin. And so this connective tissue would be our Glissen's capsule surrounding the liver, and so this adventitia is going to be continuous with a Glissen's capsule of the liver on this particular surface. Once again, looking at liver, we have our hepatic vein, lower right, sinusoids, we have our peritoneal covering, and then Glissen's capsule, the connective tissue capsule that we see here at the surface. Different stains showing a lot of the cholesterol-rich material that the hepatocytes are processing. We can see those as the white spherical structures, and we'll see them as we go up in magnification. Remember, liver cells do quite a bit of processing metabolism of cholesterols, triglycerides, lipoproteins. Once again, almost looks like goblet cells everywhere, but these are in fact the vesicles that contain this material present inside of our hepatocytes. Now, here we have somewhat of a small hepatic triad. We have our venous supply. We have our arterial supply. We can see our flattened endothelium. And here we have fairly low cuboidal cells. So this is going to be the bile duct. branch of the hepatic vein, branch of the hepatic artery. And that's going to make up our hepatic triad. Now that's somewhat of a small version. Here we have a larger version, and you can see we've duplicated some structures. We have the venous supply on the right, 2 portions of our arterial supply, and then we have 2 examples of our bile ducts here on the left. So again, another view of the periphery of the liver. We can see Glissen's capsule. We can see the serosa as we move away. Here covered with the peritoneum, this would be the adventitia if this were not present. Adventitia on this side will be reaching down toward the gallbladder. Now as we look at some electron micrographs of liver, this would be fairly easy for you to recognize these particular cells because here on these lateral borders we have our bile canaliculus, the tight fascia occludens junctions between these 2 hepatocytes. Here we have our sinusoidal endothelium, hepatic sinusoid, and then we have our space of Descemet filled with hepatic microvilli. Continuing to go up in magnification, we can again see we've got hepatic sinusoid on the left, cytoplasm of our sinusoidal capillary endothelium, space of Descemet filled with our hepatocyte microvilli. Here we have a closer view looking at the bile canaliculus. We can again see that it is also filled with microvilli. And then we can see our fascia occludens junctions between these 2 adjacent hepatocytes. We can see the very tight junctional complex is just adjacent to the canaliculus, but this is also going to run along the boundary of these 2 cells, sealing them very tightly along their lateral borders. Here we have a different view of liver and gallbladder. Liver is on the right. We have our adventitia, and then we have the gallbladder. Again, highly folded mucosa, submucosa, and a muscularis. And we'll look a little more closely at our mucosal epithelial lining. But anything associated with the liver that looks like this, we can fairly readily recognize that as gallbladder. Once again, as we're going higher and higher magnification, we can begin to appreciate that we probably have columnar epithelium. It's difficult to see if we have simple columnar, if it's pseudostratified possibly at this point. We'll take an even closer look. But certainly we have our mucosa, submucosa, and then our muscularis. And you can see the discontinuous smooth muscle in this layer before we get to our adventitia. With higher magnification, now we can plainly see our simple columnar epithelium. Now once again, ignore regions such as this and these tangential cuts right through where we have all of our nuclei, and pay particular attention to these nice runs where we see the single lined up nuclei. Here we can see that as well before we go back out of the plane of section. When you have a highly folded organ like the gallbladder, this kind of stuff is going to happen. So don't let this distract you away from seeing the true example of the epithelium that's present.